Welcome back for Chapter 7, Cellular Respiration. This is the last chapter before our next exam. Bear in mind that this is one of those weird chapters that it doesn't look like there's a whole lot to it, but there's actually a lot there to memorize. So I very strongly recommend you check out the study guides and the little summary note lectures for this, because those are going to be essential to understanding exactly what it is you're expected to know in this chapter. That said, it's not a terribly long chapter, so let's crack straight into it and see how quickly we can get through this. In the textbook, there's more images of the process of cellular respiration. The ones in the PowerPoint here are a little more simplified. It's all they're going on with here. So, cellular respiration is the process by which the release of energy from molecules is synthesized into ATP. And as we know, ATP is the energy carrier of the cell, and without it, you couldn't really do much of anything. Glucose is a very high energy molecule, and as you break it down, you are releasing energy, and then you use that energy to produce ATP. And if you go through the whole process of cellular respiration from start to finish, you get between 36 and 38 ATP per glucose molecule, which is pretty good. As you'll see later, fermentation gets you nowhere near that, so the ability to use oxygen in cellular respiration is a tremendous boon. Mind you, at one point in time, before oxygen was so abundant, the majority of organisms were or got their energy primarily via fermentation of one form or another, and oxygen killed the majority of everything living on the planet. It was one of the, I don't think that technically counts as one of the 5 or 6 great mass extinctions, depending on who you ask, but it essentially was one. But anyway, moving on. So we have our catabolic reactions, and that's a series of chemical reactions. Then you have your redox reactions, so you have reduction and oxidation. So for just remember for redox that the acronym you want to remember is OIL-REG. So oxidation is loss of electrons, and reduction is gaining electrons. So if you can keep that straight, and a redox reaction is just where you pair the two together. So aerobic respiration requires oxygen. We use the oxygen as the final electron acceptor because it is just massively electronegative. It's the most electronegative element that we can use that is biologically safe. Technically, if you could come up with some way to use chlorine in this process, because it is more electronegative, you might be able to extract more juice out of it. Put another step in there somewhere that requires a more powerful electron sink to pull everything through, and you might be able to get a little more energy out of it. But as corrosive as oxygen is, it's nothing compared to chlorine, so it's just not biologically compatible. But anyway, the final thing here is we give off carbon dioxide. And if you notice, we breathe in oxygen, we give off carbon dioxide, which means every molecule that we are inhaling of oxygen, we are exhaling a heavier carbon dioxide. This is why you were lighter in the morning than you were the night before, because the entire night you are going through cellular respiration and you are exhaling all this carbon as a byproduct. And trees take this up, and they go through photosynthesis and cellular respiration. We'll get more into that in a little bit. But they take up the CO2 and they give off oxygen. So the consequence of that is they get heavier. As a matter of fact, 90% of all of the mass in any given plant comes from the air, not from the ground or the water. It's from all of this carbon that they are essentially breathing in and adding to their own biomass. which is why when you burn them, you increase the amount of carbon dioxide in the air because they have fixed this into their system. So, interesting fact, you are made of trees, and trees are made of you. It's just this fun, I mean, like physically, the structures in your body that you built from all the glucose that you're eating from, plants making things via photosynthesis. Uh, you are made of plants, and then you're sitting here exhaling carbon dioxide, and the plants are taking that up, so the plants are made of you. It's this nice little symbiotic relationship back and forth. Uh, anyway, tangent concluded. We'll move on to. We have NAD plus and FAD. These are involved in cellular respiration and a lot of the individual reactions. They each require their own enzyme, and they utilize. Basically, just think of these as electron carriers. So your NAD plus and your FAD, they pick up electrons at specific points of the reaction, and they'll carry them from one step to another. And that's important with coupling the reactions so that the overall energy of the reactions is lowered. But moving on to these, this will be more important later. We'll move on to the stages of cellular respiration, and this is kind of the part that, at first glance, you just think, "Oh, okay, that's just a list." No, this list here is the backbone of what you need to know. So you have glycolysis, the preparatory reactions, the Krebs cycle, which we're getting away from calling things by the discoverer's name and more about what they do. So in this case, citric acid cycle is the more correct name, but on an exam I will accept either. And then the electron transport chain, which you're going to have to know, and it's not overly abundant, or it's not overly clear from the PowerPoint itself, but you need to know where each of these steps take place, what you need to get them going, and what the yield from them is. So if you know all of that, you're in pretty good shape for this chapter as far as the exam purposes are concerned. So outside the mitochondria, in the cytoplasm, you've got glycolysis. And glycolysis means to break apart sugar. Glyco, sugar, lysis, to break apart. You're breaking down the sugar, in this case, glucose. So you're taking one 6-carbon glucose molecule and you're breaking it down into 2 3-carbon molecules of pyruvate. It takes place in the cytoplasm, does not require oxygen. The energy investment is 2 ATP used to activate the glucose, and then your energy harvesting step is the oxidation of pGal, and which results in your NADH synthesis. So that's your electron carrier we talked about before. And then you also get substrate level phosphorylation, so you get 4 ATP. So your net yield here, so this is what we were talking about before, where it takes place, cytoplasm. What it requires, 2 ATP. What do you get out of it? Your net is 2 ATP, because you get 4. You spent 2, your net is, you get 2 out of it, so your net is 2. And you also get some NADH. Okay, if you understand that, you're in pretty good shape. And if I can zoom in without breaking the computer, this is just a visual representation of what you're doing. You take your glucose, split it in half, so you've got your 3-carbon molecules here. You get your direct phosphorylation, your substrate level phosphorylation to get your ATP out of it, and you end up down here at the end with your pyruvate. That's something else you need to also remember, is that at the end you have 2 pyruvate molecules. That'll be important in a minute. So your prep step, the preparatory reactions, produces the molecules that are going to enter your citric acid cycle. This occurs in the cristae of the mitochondria. So just saying mitochondria isn't enough. You need to know cristae. You take your pyruvate, your C3 molecules that have to be modified or prepped to enter the Krebs cycle, or the citric acid cycle as it's more appropriately known. And That's because the Krebs cycle only accepts 2 carbon molecules. So you take your 3-pyruvate and you convert them into 2 acetyl groups, or into 2 carbon acetyl groups. So you can see over here on the left visually what your I'm sorry, this is glycolysis again. It would make sense to put a picture of the prep reaction next to the prep, but hey, this is technically a better representation than this for what's going on in glycolysis. You got your 3 carbons split into the 2 p-gals, and that gets turned into your 2 pyruvates. You get your ATP off the end of it here, 2 from each of the pyruvates. So sure, go with that. That makes more sense. But back to prep cycle. You take your 3 carbon pyruvates, which you got here at the end of glycolysis, and you convert them into 2 carbon acetyl groups, which you then oxidize into acetyl-CoA, and you produce carbon dioxide. The acetyl group is then attached to the CoA, and it becomes acetyl-CoA. So then you remove your hydrogen atoms from pyruvate, and they're picked up by the NADH, which becomes NADH plus. Again, we're just kind of shuffling around stuff with these things. So because we got 2 pyruvate at the end of glycolysis, we do this whole prep thing twice. And so basically all you're getting out of the end of this is acetyl-CoA. You're just acetyl-CoA and NADH plus. Or I'm sorry, NADH, because you started with NAD plus, it gets transformed into NADH, and this is carrying energy for you. So 2 acetyl-CoAs per per glucose molecule. So far so good. We go to the citric acid cycle, and this requires oxygen. It's an aerobic reaction. It takes place in the cytoplasm of bacteria, but we're not bacteria, so it takes place in the mitochondrial matrix. And again, mitochondria is not enough. It's the matrix of the mitochondria. And this is a series of reactions that produce 1 ATP and CO2 per turn. And you go around twice because you've got 2 acetyl-CoAs per glucose. So for every glucose molecule, you can complete the citric acid cycle 2 times. So what you end up doing is you are completely oxidize your glucose. You've stripped all the electrons and hydrogens from it, which turns into CO2. The electrons are transferred to NAD plus and FAD, which are, again, just think of them as your shuttles, because the whole point of these is to carry that to the electron transport chain. And you get 2 ATPs per molecule of glucose through substrate level phosphorylation. Okay? So it takes place in the matrix of the mitochondria. You require oxygen. It needs acetyl-CoA because that's what you're feeding into it right here. And that came from the preparatory reactions. And the reason it's called the citric acid cycle, if we can zoom in here again without breaking anything, you're using citrate to go around the cycle here. So, and what's kind of interesting is that you take your acetyl group, your acetyl-CoA, on the citrate. The CoA does not actually attach. It goes and gets recycled to be attached to another acetyl group. And then as you're going around through here at each step, you're going from this 6-carbon monster all the way around back to your 4-carbon to the 4-carbon molecule that you bound the acetyl to, so you're regenerating itself. And then so you've got 2 of these per glucose molecule, so you can go around twice. And then at each step here, you are producing CO2, you're getting your electron carriers, and you're getting your 1 ATP over here. So the net is you got your 2 ATP and your NADH and your hydrogens, and all of those are going to be sent down to the electron transport chain. So far so good. You run your Krebs cycle twice, and this is just another I don't like this image as much, but if it makes sense to you, go for it. So you take all of that and you send it to the electron transport chain, which is located in the cristae of the mitochondria. Again, just knowing it's in the mitochondria is not sufficient. It's the cristae of the mitochondria. You get electrons from your FAD, or I'm sorry, your NADH and your FADH2, and they enter the electron transport chain. They're passed through a series of electron carriers, and each carrier is reduced and then oxidized. You get those redox reactions like we talked about before. The energy is captured and stored as a proton concentration gradient, and then the oxygen combines with hydrogen to form water, and NAD plus and FAD are recycled to pick up more electrons from glycolysis, prep reaction, and citric acid cycle. So here's the thing. All of these reactions, these individual things we've talked about, make up respiration, and they're all paired, and the products from one are often sent back to be used as components for the next. And because they're paired like this, it can be done at a remarkably high level of efficiency. So we'll look at this in a little more detail. Very simplified, this is the electron transport chain. You get your high energy electrons on your carriers here, and as you go from one carrier to the next in the chain, you're getting lower and lower energy, but you are stripping off hydrogens and forming this concentration gradient. The thing here at the end is ATP synthase, and think of this as like a hydroelectric dam. I mean, it's not exactly, but kind of. So you've got ADP and ADP and phosphates here at ATP synthase. And as hydrogens flow through this thing, it literally turns it like a turbine. And it uses that energy to bond the ATP, or the ADP to the phosphorus to produce ATP. And this is called the ATP synthase complex. And you're using chemiosmosis here through this process. That's what this concentration gradient is, running ATP synthase. And you get between 32 and 34 ATP per glucose molecule. A little more sophisticated image there. But if you can remember it from the... we broke the computer. Okay, there we go. If you can remember it from the what's going on from the simplified picture, you're in pretty good shape. So the total yield from cellular respiration, so you get 4 ATP by substrate level phosphorylation, 2 net from glycolysis, 2 from the citric acid cycle, and then you go to the electron transport chain, you get 32 to 34 ATP produced by the electron transport chain and chemiosmosis here. And then some cells have to pay to pump NADH from glycolysis into the mitochondria. That's, if I'm remembering correctly, that kind of explains the discrepancy here, because some cells are burning a bit of energy to pump the NADH around, and others don't have to go through that process, so they get a little more net out of it. This figure on the right is the one that makes the most sense to me. If you can keep this straight, you know what's happening where and what moves from place to place. This one image right here, if you can understand and explain it, that is everything you need to know for the exam from this chapter. The one on the left is a little better at kind of helping you keep straight of like what's where. It's, these are taking place in the cytoplasm, the rest are taking place in the mitochondria. The only thing is if I were you, I would print this off and list like, okay, this is taking place in the cristae, this is taking place in the matrix, and this is in the cristae, just so that way you can kind of keep straight, which is happening where. And then, of course, it also has your energy yield. So between those two, you should be in really good shape. So let's say you don't have access to oxygen, or not sufficient quantities of it. Maybe you've been running for too long, or something. You're you're up on top of a mountain. Something you've got access to, or limited access to oxygen. So what are you going to do? Well, then you ferment, and you can get some energy this way. And the thing is, it's not anywhere near as efficient as at producing energy as electron transport chain. In animal cells, you use pyruvate from glycolysis. So you go ahead and you split your glucose, and then you use that pyruvate and reduce it to lactate, which produces lactic acid. In yeasts, we generate ethyl alcohol and CO2 via fermentation. So, what this does for us, though, because you get your two lactates down here or your two alcohol in plants, it's basically the same process. You're just fermenting your pyruvate. For us, we can get a nice rapid burst of ATP, but it does not. It's only good for meeting short-term energy requirements and cannot keep pace with the demand of, say, muscles, for instance. But it is a good kind of stopgap measure to sort of give you that last little bit of oomph to keep you going in emergency situations. And I've heard this both ways about lactate being toxic to cells, but it can alter the pH. The only thing is I have seen in highly conditioned athletes, the levels of lactate actually drops in their muscles because there are additional pathways that can use this as a fuel source to kind of keep you going a little further, but that is beyond the scope of the class. That's something else you'll get into more in like biochem or something. Energy yield of fermentation. So you only get 2 ATP via your substrate level ATP synthesis, and that's a fraction of what you would get out of glycolysis. Or I'm sorry, out of the electron transport chain at the end of cellular respiration. And all they're showing you here on the right, again, if we can not break the computer, I think that's basically my catchphrase at this point. And it seems I have indeed broken it. Okay, all they're trying to show you here on the right is that there are multiple products from fermentation. So you ferment, milk, and you get this homolactic acid. You get cheese, alcohol fermentation. You get ethyl alcohol and CO2. Fun fact about that. I don't know if you all are familiar or not, but I'm going to assume that at least some of you are familiar with East Tennessee and all of these people that are obsessed with their moonshines and shiners or whatnot. Like if you ever go to Gatlinburg, you're going to see like 20 signs about, buy your moonshine here. That stuff that they sell there isn't really moonshine because it has ethanol in it. When you produce basically unaged whiskey through a still, you are producing tons of methanol in it, wood alcohol, and that's why you see like in movies they like slap the bottom of the bottle and see how many beads or bubbles form in it, because that's how you tell how much methanol's in it. Ethanol is many times more toxic per molecule than methanol. But here's the thing. We're very good at metabolizing ethanol, so we can get it out of our system quickly before it, kills you. You can still totally overdose on it, especially if you're consuming it in, shall we say, less than traditional means. But methanol is very hard for our body to process, which is why wood alcohol makes you go blind. You can't really get it out of your system quickly. So if you ever go to one of these moonshine places, just bop the bottom of the bottle. If there's a ton of bubbles that show up in it, A, that's not legal for them to be selling, and B, don't buy it because it will blind you. Uh, but anyway, so you get there's just a ton of different products, and um, this guy's feet are being destroyed by gangrene because of the different acids and acetone and stuff that's being produced by that fermentation. It's a very adaptable system that can be used on multiple things under different conditions to produce multiple products. Is the whole point of that. And anyway, tangents aside, this is not anywhere near as bad of a chapter as it looks. You just need to know the specific things I told you. What happened? Like, where does a thing happen? What does it need to make it happen? What do you get out of it? And then what goes to the next step, basically. And if you know all of that, you're good to go. The exam is next time in class. If you have any questions, comments, concerns, let me know, and I will see you next time.